Hello, everyone. Thank you for attending today's webinar, Implications of Historical Control Studies for Rare Disease Products, presented by Roe. I'm Ricardo Castano, and I'll be moderating this webinar. Our speakers today are Rob Wolfson, Chief Strategist of Biostatistics and Standards for Regulatory Submissions at Roe, and Kevin Barber, PhD, Vice President of Regulatory Strategy and Submission at Roe. You can read their full bios on the left side of your screen by selecting the Speakers tab. Just a few technical notes before we begin. If you'd like to download the slide deck, please click the Handouts tab button on the left side of your screen. This webcast is being streamed to your computer, so there is no dial-in number. For the best audio quality, please make sure your volume is up. This webcast is being streamed to your computer, so there is no dial-in number. For the best audio quality, please make sure your volume is up. This webinar is being recorded and will be available on demand within 24 hours after the event. Time permitting, we will follow the presentations with a Q&A session. Please submit your questions using the Questions and Answers tab on the left side of your screen. Okay, now let's begin. Kevin, please go ahead.
All right, well, thank you, and thanks very much for all of you for joining us this afternoon. Here's a quick outline of our presentation for reference when you download the slides. We're also going to be providing some links to some references at the end of the presentation that you'll have as part of the downloadable file for your future reference. So by way of introduction, as noted here, pharma and biotech companies are focusing more and more nowadays on rare diseases and development of therapies for orphan indications. And given the circumstances associated with trying to investigate therapies in these rare disease populations, sponsors really have to carefully consider what's feasible in the design of clinical studies. And this leads them to consider more non-standard study designs that could include historical control groups to assess the efficacy of a new intervention. So in our presentation today, we'd like to review some of the factors involved in the use of external or historical controls in a clinical study, along with points that you need to keep in mind regarding analysis and interpretation of the data from such studies. And we'd also like to briefly discuss the role of these studies to support marketing applications for rare disease products to treat rare diseases. So I'd like to start out just with some brief background on rare diseases and historical control studies. I think, of course, as we all know, back in the early 1980s, the Orphan Drug Act defined rare diseases as those affecting fewer than 200,000 people in the United States. So given the rarity of these diseases with such small patient populations, along with the nature of, in particular, the more serious of these rare diseases, there are a number of critical elements that you need to keep in mind for a successful clinical development program. First and foremost is having a clear and detailed understanding of the natural history of the disease. This is crucial, and really the other elements listed here depend upon that. You also need to have a sufficient understanding of your drug's mechanism of action in treating the disease to help guide you in designing your study and selecting endpoints. And of course, overall, based on the natural history and the progression of patients with the disease, you need to be able to select clinically meaningful endpoints. And ultimately, to get the drug approved, you need to understand the regulatory requirements for the amount of evidence that FDA and other regulatory authorities are going to need in order to approve the product with appropriate demonstration of safety and effectiveness. And that last point really results in a major challenge in developing products for rare diseases. Basically, given the extremely small patient populations, coupled with the potential lack of therapy or perhaps adequate standard of care, trying to design studies that use concurrent controls such as a placebo arm become impractical or potentially unethical. And this drives drug developers to consider historical controls as an alternative in these circumstances. So in a little bit, we'll review the definitions of historical control trials. But first, I want to turn it over to Rob to talk a bit about sources and use of historical data.
Thank you, Kevin.
Rob?
Yep. Thank you, Kevin. Kevin provided the motivation for the historical control study design. And I'm going to spend just a few minutes talking about the various sources that are typically used to inform external control groups. First, though, as background, and Kevin will discuss this in more detail later, but to provide some reference, generally speaking, historical control studies include patients with the same disease who were studied at an earlier time. And this cohort or group of patients form a reference or a control group against which a treatment arm and the results of that treatment can be compared. The sources of those data are numerous, and I will discuss a few of them in just a moment. Broadly speaking, the sources of data fall into observational study type data, medical records, and the most robust form of data for the purposes of historical control studies, a control group or treatment arm from a previously conducted clinical trial. I'll discuss in the coming slides the limitations and benefits of these various sources in the historical control study design context. I'll briefly cover how these data are used and our familiarity with using these type of data in post-marketing surveillance activities, and then transitioning to a setting which is a little bit more new and where the history is a little less rich in terms of trying to assess the effectiveness of a proposed therapy in a rare disease setting. I will then focus in on how these data have been used in clinical trials. Part of this comes from our experience in practice, and this will be the focus of the remainder of the discussion that I step through and that Kevin steps through as well. So there are numerous sources of historical data that can be used to inform externally controlled studies. I've laid out a few here. These are described in more detail in the FDA's framework for its real-world evidence program. Generally speaking, I've divided these up into three buckets, and this is the way I will discuss them going forward. So one is the bucket of real-world data. The second bucket is clinical trial data, often data coming from a randomized controlled trial, but not necessarily. And then third, a natural history data set used to understand the natural course of disease or course of disease under a particular standard of care. So let me talk about these three buckets quickly in terms of limitations and benefits. First, real-world data. Real-world data are data relating to patient health status and or the delivery of healthcare routinely collected from a variety of sources. And again, I've laid them out here. I think we're all generally familiar with what these are, but let me talk about them in the context of considering them for a historical control study. So electronic health records, while certainly rich in terms of patient information, and I won't cover that. I think we all have some idea of what's contained in electronic health records, do offer numerous challenges that I'll discuss in more detail later on, but that I'll briefly bring up here. So in terms of using these data to help inform a historical control study and to form a reasonable comparator group, we typically run into issues like lack of standardization in collection techniques, inconsistent laboratory assessments across organizations or hospitals, incomplete data, differing systems being used to collect those data, which lead to a lack of interoperability, and so on. So while a rich source of information, their overall utility is oftentimes limited. The second source of data falling under the real-world data bucket are medical claims and billing information. So while the information here are more limited than patient information contained in an electronic health record, they do offer numerous benefits, including highly standardized data in most cases. So medical diagnoses and things like that tend to be harmonized and standardized and make extraction of data, interpretation of data a little bit easier. The third bucket are disease registries or data registries. These are observational studies that collect patient information and typically in some sort of uniform way. This is usually a targeted population where outcomes are assessed for a predefined purpose. So these oftentimes include many, many thousands of patients and many records of patient data. Usually many, many variables, including covariates, are collected. And again, this makes for a very rich source of potential data to be used in support of an external control group in this type of study design. However, any of you who have explored disease registries or patient registries knows that data quality is often an issue. Consistent reporting can still be an issue, and these data sets are often hampered by missing data, as well as other issues one typically sees in observational studies. Next bucket I'll briefly touch on are prior clinical trial data, and I'll really just specifically briefly talk about clinical trial data in the randomized controlled trial setting. This tends to be a very robust and rich source of information to be used in informing and defining an external control group, in particular, patient-level information. So where we have patient-level information, various statistical techniques can be employed to try to minimize bias and other issues that may arise in this setting where we don't have the protections of the randomized controlled clinical trial. There are, of course, situations where results of those studies are used to help inform studies of this type, but I'm not going to spend any time on that particular scenario. The third and final bucket that I'll talk about is our natural history data. Again, these are observational studies intended to track the course of a disease. These can either be prospective or retrospective, but again, are very rich sources of information for external control group definition, but suffer from the same types of issues that registry type data often suffer from. I think we're all familiar, or at least most of us are going to be familiar with efforts of the FDA and other regulatory bodies to use real-world data to generate real-world evidence of safety. And again, this is discussed in detail in the FDA's framework for real-world evidence. But I think that we all likely have some experience with using data collected in electronic health records or from registries, spontaneous reporting, to get some sense and to evaluate the overall safety profile of a product, typically in a post-marketing type situation. And in this setting, FDA, sponsors, contract research organizations have developed extensive experience evaluating data quality, data reliability, utility of data, things like that in this setting. There is, however, less precedent in establishing effectiveness of a particular treatment or proposed therapy, and evaluating these data for reliability, quality control, relevance, and so on. And so I'll discuss that in a little bit more detail. I'm going to take the next two slides to talk about the uses of historical data in clinical studies of rare diseases. And I'm going to divide this up into a couple of buckets. And again, this is consistent with the way the FDA thinks about and presents their thinking on this topic. First, how historical data are used in the design of clinical trials. I'll discuss a few scenarios shown on this slide here. And then next, I'll discuss how historical data are incorporated into a study itself to inform conclusions related to safety and effectiveness. And again, this is where most of our discussion going forward will be. This is sort of the colloquial historical control study. So there is a history of using historical controlled data in establishing effectiveness. And historical data have routinely been used, and one could argue, though not possibly quite enough, to improve efficiency in trial design. Let me talk about four ways in which this is done. First, historical data has been used, and we've done this in practice extensively, to generate hypotheses for further exploration in clinical studies or as part of a clinical development program. So we routinely explore registries of data, natural history studies, and so on to generate hypotheses which are then studied as a part of a clinical development program. Statisticians, clinical experts, et cetera, design protocols around these hypotheses. Again, we use the historical data to generate those hypotheses. Next, historical data has been used to identify drug development tools, surrogates for disease progression, biomarkers, patient-reported outcome development, other covariates important to progression or slowing of disease. So, a typical method by which historical data are used to inform clinical studies. Third, trial feasibility. And for those of you that work closely with clinical folks, this will, of course, ring true. Historical data is often used to help formulate, define inclusion-exclusion criteria, geography of study, locales of study, potential sites, enrollment projections, things like that. And then the last example that I'll provide, of historical data being used to inform clinical studies is when it's used as a prognostic indicator to identify patients that may be most likely to benefit from a proposed therapy. So a typical example here might be an enriched enrollment type study design. We may be able to, ahead of enrollment or screening or post-screening, identify patients who are most likely to benefit from the therapy and enroll those patients as a part of our clinical trial and in answering our particular research question of interest. So that was the first step, how historical data are used in improving the efficiency of sort of standard clinical study designs. Now I'm going to focus in more so on the topic of today, which is how data are used to actually help us make determinations related to the effective treatment in clinical trials. So these are situations where data are incorporated into or are the sole source of data in a clinical trial, and these data are used to support effectiveness claims. Again, four buckets here that I will quickly step through. First are natural history studies, which I described briefly. I will not go over them again. These can be retrospective or prospective. Both have pros and cons with respect to time, timing of collection, readout of results, completeness of data collection, and so on and so forth, but that's really outside the scope of this discussion. But natural history studies, of course, are a form of historical data being used in this type of study. Second, we can incorporate historical data into a predefined trial decision-making process. So there are numerous cases cited in the literature where prior information collected from an earlier study is used to inform stopping boundaries, pruning decisions, dose-related decisions, and the small amounts of data that are collected in an ongoing study are then supplemented with this data to help inform these decisions in some predefined way. Third way in which historical data are actually incorporated into a clinical trial is via an amalgamation of the data collected from an external control or from an external group, historical data, and incorporating that into the current clinical study. So, for example, data collected in a control arm in an ongoing study is amalgamated with historical data, and then decisions with respect to effectiveness are made, assuming that the data in the control arm and the external arm are sufficiently similar, and there are statistical methods to assess this. But this can lead to reduced costs, obviously, a higher number of folks being enrolled in a treatment arm, and of course offers numerous other benefits, risks as well. And then the last group is where historical data are actually used as the external comparator itself. And most notably, these are non-concurrent external comparators in the colloquial sense. So I will get into further discussion about that here. Kevin will walk you through the various regulatory definitions of historical control study, in particular focusing on this external comparator non-concurrent setting. He'll also provide the regulatory framework for establishing the effectiveness of a particular proposed therapy via studies of this type.
All right. Thank you, Rob. So yeah, as Rob noted, we talked briefly about historical data. So let's review, as Rob said, the definitions of historical control and historical controlled studies briefly that show up in regulations and in guidance. So we can start basically by quoting chapter and verse from the Code of Federal Regulations, 21 CFR 314, Section 126. And this is the section in the regulations that defines adequate and well-controlled studies, including the definitions of 4 different types of concurrent controls plus historical control. I'm not going to read this verbatim, but a couple of things to note. First off, historical control was included in this section of the regulations from the get-go, from the first day it was written several decades ago. So from the beginning, it's been recognized as a viable control for an adequate and well-controlled study. And as defined here, with historical control, your active treatment is compared with data and experience derived from documented natural history or from active treatment in comparable patients or populations. And very important to note, as highlighted here, historical controls are reserved for special circumstances, which we'll talk about in just a moment. Historical control studies and historical control is also defined in FDA's recent 2019 rare disease guidance, which I've quoted here. And this is actually defined to a somewhat, I think, more explicit level of detail, noting that in these studies, all patients receive the investigational treatment and there is no randomization to a concurrent comparator group, which means that the comparator data, as Rob has been describing, comes from outside of the study. This guidance also defines to some extent the special circumstances in which use of this type of control is appropriate. Generally, we're talking about serious rare diseases with an unmet medical need. And one other place that defines this is the ICH E10 guidance on the choice of control group for clinical trials from 2001. And this guidance provides the definition of an external control and notes that the external control can be a historical control consisting of a group of patients treated at an earlier time. So with these sort of definitions in mind, let's take these various presentations of the definition of a historical control and pull them together. And here's basically a working definition of a historical control trial. It's a trial in which there's no randomization to a concurrent comparator group, and all enrolled patients receive the investigational drug, and any conclusions about the drug are drawn by comparison with the non-concurrent historical control population treated at an earlier time. And also note that these historical control studies are basically a subset of external controls, which are defined as a control consisting of a group of patients treated in another setting external to your given study. So as noted in ICH E10 and in other guidances, you can basically lump historical controls into 2 types, a population of patients with the same disease observed at an earlier time and patients as their own control. The first one here is what I would typically consider to be the more common approach to historical control, but the second can have utility as a control as long as you have enough information to reasonably extrapolate from baseline for each patient so that you can get an estimate of what would have happened without treatment. So this means, of course, that the natural disease progression must be very well characterized to do that extrapolation. Okay, so we've talked about the sources of historical data and the definitions and types of historical control. So I just want to talk very briefly about the regulatory framework before turning it back over to Rob to talk in more detail about some of the critical considerations for historical control studies. So as FDA has often noted, the Orphan Drug Act of 1983 did not create a separate statutory or regulatory standard for the evidence required to demonstrate effectiveness of drugs to treat rare diseases. In other words, FDA still requires, quote, substantial evidence for the drug and its intended use to demonstrate safety and efficacy for the product in the treatment of a rare disease. So to develop substantial evidence, what are we talking about? I think all of you are probably familiar with this, and this is basically just a review, but let's step through it. The standard for FDA to approve a drug is substantial evidence of effectiveness from appropriate, adequate, and well-controlled studies. And FDA's interpretation, grounded both in science as well as in the statutory language, is that at least 2 adequate and well-controlled studies are necessary to provide independent substantiation and confirmation of the effectiveness of the product in treating the rare disease, or in treating the disease, rather. And the gold standard for such studies consists of randomized, concurrent, controlled clinical studies. Now, per both the Act as well as regulations and guidance, FDA does have statutory authority and administrative flexibility on a case-by-case basis to accept less than this gold standard that we just described. So under certain circumstances, and we're primarily talking here today about serious rare diseases where a second confirmatory study is impractical or unethical, FDA has demonstrated flexibility by accepting a single adequate and well-controlled study. This is based both on the Food and Drug Administration Modernization Act, Section 115 from 1998, which states that FDA can accept a single study plus appropriate confirmatory evidence, as well as FDA's evidence of effectiveness guidance is listed here. And beyond the single study question, FDA has approved drugs to treat rare diseases based on historical controlled studies, but as noted in the definition of an adequate, well-controlled study that I quoted earlier, historical control is limited to special circumstances. So what constitutes special circumstances such that historical control is acceptable? The first 2 conditions listed here in this slide are absolutely paramount. When you're developing a drug to treat a serious condition with an unmet medical need, and concurrent controlled studies are impractical or unethical. And as noted here, we're talking about diseases with high predictable mortality or progressive morbidity, and you need to have very solid information about the disease course so that you can measure it objectively. So again, these are the primary points about the special circumstances under which FDA would consider historical control to be appropriate. So one final comment for me, and then I'll turn it over to Rob to talk about the critical aspects of historical control studies in more detail. But this slide presents the conditions that FDA considers when reviewing the results of a historical controlled study to gauge whether the study has provided substantial evidence of effectiveness. Rob's going to talk about some of these in more detail, but the key messages here are that the historical control group is sufficiently similar to the active treatment group in a number of factors, and the study must provide compelling evidence that the investigational treatment changed the progression of the disease. So I'm going to turn it back over to you, Rob.
Thank you, Kevin. So I'm going to step through several factors to consider when designing a non-concurrent, externally controlled, historical, data-driven study. One of the first factors to consider is whether or not you've engaged a statistician, and it is quite important that you do so early on in the design and consideration process. I'm going to step through some basic guidelines based on our experience, which is a combination of the practical application of FDA guidance, our FDA interactions, as well as a couple of very rudimentary statistical concepts. We'll focus on a few areas. There are 3 in total here. One is evaluating the external data for reliability and usability. I will cover that topic a little bit. 2, I'll discuss the meaning of what an adequate control group is. While this is clear in a randomized control trial setting, regardless of the question being addressed, whether it's a superiority-related question, a non-inferiority-related question, it may not be quite as clear in a historical control setting, and I'll discuss that briefly. 3rd, I will discuss an evaluation of the external control group, in particular the factors that we consider in assessing similarities and dissimilarities between a proposed external control group and a proposed treatment group. Thereafter, I'll move into a brief discussion of advantages and disadvantages of this type of design. This will really serve as a summary of those three topics I just discussed. And then finally, let's assume for a moment we've completed a non-concurrent historical control type study, externally controlled. What are the factors to consider? The guidelines that both FDA and we have used for interpreting historical control studies, and I'll spend just a moment putting these in context, in particular with the context being a more typical randomized controlled trial. So we've put together a handful of general guidelines that we use when designing a historical control study. And there are 5 here. And there are other considerations and factors one might want to consider, and I'll touch on a couple of those. But this is generally the roadmap that we follow in designing one of these studies. Of course, there are details that we're not discussing here. But first, as with any clinical study, is to define the research, regulatory, clinical, scientific question that needs to be addressed and answered. And of course, this is foundational for any clinical trial and won't be discussed further here. Second question is whether or not a historical study design is appropriate. And Kevin laid out some of those factors 2 slides ago. But in short, we weigh and discuss with sponsors ethical considerations, whether or not there's a lack of equipoise between the treatment groups, proposed treatment groups, the feasibility of a randomized controlled trial, any enrollment difficulties that we might be able to overcome in the preferred randomized controlled trial setting, whether or not this is a disease with a well-characterized progression pattern. Those that are highly variable are less applicable to a historical control setting. I'll discuss that a little bit later. It is good to engage with regulators in advance to discuss proposed historical control study type designs. And this is based on some of the information that Kevin provided a few slides ago. Third is to evaluate the available sources of data, and I covered this in my earlier section. So whether the data are from medical records or medical claims records, electronic health records, registry, a prior clinical trial, some evaluation of those data should be conducted early on to determine whether or not they're even usable or suitable for such a study. Next will be I'll discuss, and our next step in this process is to evaluate the robustness, the utility of the actual data. So let's assume for a moment we've overcome the hurdle of the data being usable or not. Let's assume that it is. We now ask several questions. Are the data fit for purpose? Are they going to answer the questions we need to answer? Is there exposure information, access to covariates that we're going to need for analysis and help in identifying the true effective treatment? How standardized are the laboratory data that were collected? So on and so forth. So I will discuss that in a little bit more detail, particularly in a standards-related context. Fifth will be an evaluation of the similarity of the proposed external control group and the planned treatment group. And this, quite frankly, is perhaps the most important consideration in evaluating the utility of a particular external control group and the viability of a historical control study design. And speaking as a statistician, large dissimilarities can impact interpretation and can impact interpretation of results and the estimates of treatment effect sometimes greatly. And there are cases where we can't overcome some of these issues with statistical methodology, and I'll discuss that in a little bit. But really what we're dealing with here is bias, a phrase everybody's familiar with, in particular selection bias. There are other forms of bias that can enter in, but in particular selection bias. We don't have randomization to protect us and ensure that folks were randomly assigned to treatment. What's not on this list, and certainly should be considered and will be discussed in the interpretation section, are the methods used to compare the groups. What are the appropriate methods? Are there appropriate methods? Those 2 questions, again, to be discussed in the interpretation section. I'm going to focus on those last 2 items on the prior slide. So let me jump to evaluating the utility of the external data. And I've run through what some of those common sources are. A key topic that you've probably heard me continuously bring up is standardization of those data. And I'm going to spend just a moment of time on that and its role in evaluating the utility of an external data source to support a historical control study. So I've noted several of the challenges of non-standardized data, including collection techniques, laboratories, completeness, consistent coding, different systems, and so on and so forth, different formats. The list gets quite lengthy. A lot of these issues have been addressed in CDISC standards development, an acronym I'm sure most of you are familiar with. These very issues that I'm talking about in external data sources were previously seen in clinical trials conducted decades ago, and again are apparent in our review and consideration of external data and external control groups. So the use or non-use of these data standards is an important consideration and one that shouldn't be overlooked, and a statistician can help evaluate data in light of CDISC-related standards and the utility they provide. If CDISC standards are used, interoperability is greatly improved, efficiencies are gained, and certainly helps in setting up, evaluating, and analyzing a historical control study. At the design stage, it's important to evaluate the availability or utility of a potential external control group in light of the FDA's definition of an adequate control group. So I won't read this here, but let me provide what I think the two key takeaways are here, and I think they're self-evident if one reads this. One is that the FDA recognizes that which we're all aware of, which is that estimates of treatment effect can be influenced or confounded by factors other than the treatment itself. And this makes interpretation awfully difficult. Second, we need to be able to discriminate the true effect of treatment from the effect of these other factors. So these systematic sources of error, while generally reduced and controlled in a blinded, randomized controlled trial setting, are more difficult to control, and there may in fact be no control over them in an externally controlled study. So a key question to overcome these two issues here, or meet these two requirements, is how similar are the planned treatment group and planned external control group? And this similarity or dissimilarity is a foundation of an interpretable historical control study. So put simply, the effective treatment needs to be capable of estimation in an unbiased manner. I will note that where we're not sure that the estimate is unbiased, we have had experiences where large treatment effects relative to the sources of error or bias may result in a favorable interpretation of the study results. But the general default position, as Kevin presented earlier, is that estimation needs to be unbiased, an unbiased estimate of that true treatment effect. So randomized controlled trials and the attributes that these study designs offer, blinding, randomization, protect us from some of these sources of bias. These features are no longer there, and we need to consider this as we assess our proposed external control group. So without randomization, how do we assess the utility of a proposed external control group? Again, we assess similarity. In short, this is laid out in a couple of different FDA guidances. The one in particular that we're pulling this from is noted at the bottom, but we have found these to be true in practice, routinely evaluated not only by our clinical experts, but also by FDA regulators. So the key question in assessing some of the factors I'll step through here in a moment is how similar are the groups, and what justifies our belief that these groups are substantially similar and suitable for comparison. So the key factors, as discussed in this guidance, again, are the same factors we commonly hear and discuss during product review: state or severity of the disease and differences between the groups, differences in disease assessment techniques or terminology, any differences in baseline characteristics, standard of care, or methods of data collection. And differences in any of these can introduce a systematic bias that would concern us in a randomized control trial setting. So let me quickly step through each of these. In short, we are concerned about the similarity in disease state or disease status between the two groups, and I've laid out what some of those factors are. And systematic differences can impact the estimates of treatment effect, and it's possible that effects of treatment are over or underestimated based on these systematic differences. So an obvious example is patients with exclusively severe disease are compared with patients with exclusively mild disease. and the group's treatment and external line up perfectly relative to those diagnoses. Estimates related to effective treatment are likely impacted by severity, and more specifically, the difference in that severity between the groups. The estimates are no longer unbiased. So I will discuss this in just a few moments, but patient-level data can be very helpful in trying to pull apart and identify the effective treatment, but techniques may not be able to overcome dramatic differences. I'll discuss a couple of those techniques in just a couple of slides. Another set of factors to assess is the similarity in baseline characteristics, disease assessment techniques, and standard of care. So baseline characteristics are an obvious consideration, and I'm not going to spend any time on those. Next is that disease assessment techniques and differences can have a meaningful impact on our ability to determine or to interpret treatment-related estimates. It is common for disease assessment techniques to change over time, and we see this frequently as we are considering and designing historical control studies. And these differences can impact the type of patient in the external control group and in the proposed treatment group. Standard of care, of course, changes and generally improves over time. In addition, there are generally differences in countries, cities, individual clinical sites. This, of course, is the motivation for the multi-center trial design. And groups in disease progression can be impacted by time-driven changes in standard of care, site-related differences, standard of care, and so on. And as the FDA notes, and we found this to be true, it's usually helpful for there to be a close temporal relationship between the external control data set and the time those folks were treated and the proposed treatment group. Lastly is the similarity of data and endpoint collection, and this one is going to be near and dear to statisticians' hearts. So while this is often overlooked in the design of historical control studies, this is a common issue that plagues the interpretability of such studies. So even in cases where similar assessment tools were used, we look for similarity in timing of collection, the intervals of collection, objective measurements, things like that. So some of the common issues, I'll just hit on a couple, are that the assessment methodology may be different or changed between the two groups. We may not collect all the data variables in the external group that we would like to collect in the treatment group. A common one is that medical terminology has changed over time. It becomes inconsistent, and ferreting out meaningful outcomes, comparable outcomes, can be different or can be difficult. So let me summarize here what I've just presented. Some of the advantages and disadvantages of a historical control study. So we've considered as much as we can. We think we've laid out a design. Let's take one last look at the advantages and disadvantages of such a design. So advantages, as Kevin pointed out, everybody gets the drug. We may be able to overcome ethical considerations. We may be able to reduce costs relative to a randomized controlled trial. And we may even now be able to conduct a trial that otherwise would not have been conducted in a randomized controlled trial setting. Some of the disadvantages: no randomization, no blinding. We may not have a perfect set of data from which to extract our external control group, and the interpretation of results can be quite difficult. And I'm going to discuss this now in the next handful of slides. So let's assume for a moment that the study is completed. How does one interpret the results from an externally controlled study? We'll provide two guidelines for interpreting study results, and then offer a set of factors to be considered as one interprets treatment estimates. So first, let me go to the guidelines. Our two suggestions from our practice is as follows. One, compare the treatment group and the external comparator using several of the factors that I described in the prior slide. So in other words, how different are the groups? Are there systematic differences between the groups that could impact or bias the estimated effective treatment? And I'll step through those again very, very quickly. Secondly, it is wise to consider the methodology that was used to generate the estimated treatment effect. Did it involve all comers? So everybody that was in an external control group data set was compared with everybody in the treatment group? Or was there some sort of cohort type matching or other methodology that allows us to to make a clearer comparison between the groups? Details are beyond the scope of what I'm going to talk about here, but a couple of the more common methods I'll bring up in one particular slide here. So let's review once more the factors that can systematically differ between the groups. As I mentioned before, based on the FDA's language and based on our practice, the goal, of course, is to estimate the effective treatment separate from other factors that could have influenced outcomes. And we've discussed several of those factors laid out here. One I did want to bring up that I've not previously brought up is patient attrition, in other words, missing data. And this has been given much attention in the randomized controlled trial setting and in other study designs. And in particular, the release of the National Academy of Sciences document on missing data summarized very nicely the impacts of missing data on estimates of treatment effect, in particular in the randomized controlled trial setting. And many of the same issues apply here in the externally controlled setting and should be considered. So differential dropout between the groups can impact and bias estimated effects of treatment. So something to be considered in the interpretation of historical control study results. Let's quickly talk about a couple of matching methods that can be used to reduce potential bias and control for confounding factors. So as I mentioned, considerations should be made regarding the method used to compare groups and estimate treatment effects. Treatment assignment is not random in this setting. As you know, there is no random assignment of treatment, and the likelihood of a systematic difference between groups is very real and needs to be considered. However, there are methods that exist that attempt, at least in some respects, to do something like mimic randomization. They can limit or remove bias and account for some confounding factors. And two of those methods are direct or exact matching and then propensity score matching. And I will note for completeness that propensity score matching can be used in other ways, not just matching, but that's the only manner in which I'll talk about it here. So at a very high level, the goal of both of these approaches is to account for the fact that treatment assignment was not random and match groups of patients based on key variables of interest. And the hope is that in doing this, there's some balance struck between the groups, and we can make a comparison that is unbiased and meaningful with respect to the effect of treatment. So with direct matching, patients are matched as the word describes, exactly or directly. However, this can get complex, as you can imagine. You can quickly go from matching on just one variable to 10 or 15 variables, and you can start losing patients, and this has deleterious effects on our ability to interpret study results. As a result, propensity score matching methodology was introduced, and a large number of variables can be used to match on a single score. There are downsides to this approach, but it is appealing in the sense that numerous, possibly important confounding variables can be included and patients matched and balanced accordingly. To give you some point of reference here, in a 1-to-1 randomized controlled trial, a propensity score for any given patient would be 50%, so you can see why this is an attractive approach. Methods such as these, and there are others, can help produce unbiased estimates of a true treatment effect. So let me quickly tie this out to randomized controlled trials. Randomized controlled trials offer protections from certain sources of bias, and we know that the same cannot be said of historical control studies, most notably the absence of the random assignment of treatments. So regardless of the methodology used, how do we interpret the results? In short, this is a balancing act, and typically what we're balancing here is a point estimate and a confidence interval of a particular treatment effect in an external control group relative to a point estimate and a confidence interval in a treatment group, so external control group and treatment group. And what we're balancing here is the size of that effect, the width of those confidence interval, against other things that we feel could possibly influence or confound the estimates that we're coming up with. So, for example, how predictable is the disease in its progression? Are there potential biases that, despite whatever methodology was used in terms of matching and so forth, that we can't or are unable to account for? How influential are those potential sources of bias? The temporal relationship of the outcome to the intervention. So as more time elapses between intervention and the outcome, we possibly have less certainty around the estimate or the impact of treatment in that setting. And then finally, we look at the actual outcome itself. So objective outcomes are favorable. Something like mortality is easy to report, certainly not a desirable outcome, but very easy to report, clear, objective, and that is favored in this setting over something more subjective. There is some precedent for regulatory approvals of rare disease products using historical control studies as the primary source of evidence of effectiveness, and Kevin will review at least one of those approvals. So, Kevin?
Thank you, Rob. Yeah, I think we have 4 examples in our slide deck. I think in the interest of time, we do want to allow some time for questions. So I might just go through this first one in a bit of detail, and then move through our summary. But the first of these examples is Protominid, which was approved just last August under the so-called LPAD pathway, which is a limited population for anti-infective drugs pathway that FDA has in place. And it was approved for treating basically extensively resistant, drug-resistant tuberculosis. So this is an example of an approach for historical control that used both a matched historical cohort as well as tuberculosis outcomes reported in the literature. It was an open-label study in 109 patients with resistant TB. And as I mentioned, the historical control was a matched cohort as well as the literature. So it's a little bit interesting in that regard. The primary endpoint is defined here with respect to treatment failure, and sort of cutting to the chase, the rates of favorable outcomes for 6 months, at 6 months rather, following end of treatment was 89% in the open label active treatment study. And this compares with a pre-specified historical control rate of 50%, as well as in the matched historical control, the favorable outcome was 11%. So a bit of difference. And FDA noted where I have the bullet that says lower confidence interval or lower confidence limit for success exceeded. The FDA actually had the word greatly exceeded the pre-specified control rate. So again, this is one that was based on a basically a case matched as well as a literature approach. Quickly going through the other ones, I have this example for Privigen here as one that you can take a look at that used the placebo arm of a previous study as the historical control. The next two, Brineura and Kanuma, both used retrospective natural history studies as their control arms, and you can take a look at that. I will note that Kanuma actually was approved on the basis of 2 studies, not just one, so a historical control plus a randomized study. As I mentioned at the beginning, we wanted to provide some links to the references that we've discussed in this presentation, so they're presented here on this slide. And then Rob, very quickly, I want to turn it over to you for summary, and then we can move on to some questions.
Thanks, Kevin.
Rob?
So in summary, thanks, Kevin. In summary, product development in the rare disease setting does present numerous challenges, and the use of randomized, blinded, concurrent control studies may be impractical, unethical, not feasible in this particular setting. Recognizing these challenges, Kevin has laid out nicely the FDA's regulatory framework for developing rare disease products using methods other than the randomized controlled trial to estimate the effect of treatment, and these include historical control studies that we've spent our time talking about. So while there are numerous benefits to historical control studies, the regulatory and scientific burden to produce an unbiased estimate of the effect of treatment is not lowered. And as a result, careful consideration does need to be paid to the choice of external control group, the similarities of that group to the treatment group, and the quality of the external data, all points we've discussed in detail. As Kevin just wrapped up, regulatory precedent does exist for approving products using these types of studies in the rare disease setting, and rare disease and orphan type product submissions are becoming more prevalent to FDA. Oh, there we go.
All right. So with that, it looks like we're good to move on to the Q&A. Remember, everyone, there is still time to submit questions using the Q&A tab to the left side of the screen. We have a lot of great questions today, and we will try to get to as many as possible, but we will, if we won't be able to get to them all today, but we will try our best to get back to everyone who submitted personally after the webinar. So let's see this first question here. This is for either of you guys. When is it unethical to do a concurrent control arm like placebo or active standard of care?
Yeah, this is Kevin. I'll I'll answer this quickly. Obviously, this is case by case and perhaps a bit subjective, but again, we're dealing with serious, life-threatening rare diseases with an unmet medical need as sort of the situations and special circumstances. And often there is either no treatment at all available, or the current standard of care is poor or very poor, and of course the prognosis and outcome for the patients is poor as well. So these are sort of the basic conditions for considering whether it's unethical to do a placebo arm, for example, or whether to rely on the standard of care. And another factor is, has your product demonstrated, even though you haven't done a so-called adequate and well-controlled study to support approval, has your product demonstrated the potential to demonstrate efficacy? And if so, you may be in a situation where it would be considered not ethical to include a concurrent control arm of either placebo or standard of care, and make the argument that it's most appropriate to use an external control such as a historical control.
All right, thank you, guys. Let's see. The next question asks, are you aware of situations where a health authority required a natural history study?
This is Kevin. I guess I'll start, and maybe Rob, you could chime in as well on your level of awareness. I'm not aware of a situation in which FDA. Well, I guess it's almost implicit. FDA and other agencies for these rare diseases consider natural history studies basically to be essential to inform development programs and to assist them in their regulatory review and being in a position to make approval decisions. So while FDA can recommend and suggest and so on, I think the experience around, quote, requiring one is probably implicit if FDA says, look, there's not enough information available for us to agree that your study design is appropriate, or they're in a position where they can't approve a product unless you go back and develop enough natural history information to support it. So at a high level, I'm not aware where FDA said you must do this, but it is something that they consider to be essential. And Rob, I'll see if you have anything to add to that.
No, I don't. Your experience is consistent with mine, Kevin. I think that's a good summary.
Oh, okay. All right. Thanks.
All right. Thanks, guys. So let's see. This next question, it asks, what is the best or most optimal source of historical control data?
Rob, you want to?
Yeah, I'll take that one. So I'm not sure that there is a best or optimal source of historical control data. I will speak to our experience, though, in terms of usability. When evaluating various sources of external control data, we go through the steps that I described in one of the earlier slides. With respect to whether or not the data are fit for purpose, whether or not the data are particularly standardized, usable in a consistent format, and so on. In that respect, we have found that patient-level data coming from a clinical trial, hopefully a recently completed clinical trial, tends to satisfy most of the considerations we have on the usability, utility of the data. When it comes to evaluating the data source in the external control group for comparability, interpretation, matching related potential, again, patient-level data coming from a completed clinical trial, in our experience, tends to be best. This is for a few reasons. One, data were collected under a protocol, so it is clear what assessments are made, when they're made, how they're made, and that can be used in informing how we select our treatment group, the protocol design around that treatment group, and so on. Next, we know that the set of patients that we're dealing with in data coming from a completed, previously completed clinical trial were selected with specific inclusion and exclusion criteria. So some of the covariates and the confounders that I've spent a great deal of time discussing, whether they're baseline demographics or disease severity and so on, are things that we know have been considered in the enrollment of patients into that particular data set. So for those reasons, while I don't know that there's an optimal source of historical control data, those data coming from a completed clinical trial tend to be most usable in our experience.
All right, thank you so much. And unfortunately, that will have to be our final question for today. We had a lot of great ones today. Like I said, we couldn't get to them all, but we will try our best to get back to everyone who submitted personally after the webinar. With that, I'd like to thank you all for attending this Fierce Markets webinar and for submitting so many great questions. I'd like to thank our speakers for participating, and I'd like to thank Ro for presenting today's webinar. Remember, this webinar has been recorded. You will be able to access the recording within 24 hours using the same audience link that was sent to you earlier. Thank you again for joining, and we look forward to seeing you at future events.